Okay, welcome back friends. In this video we'll be talking about primer designing. Okay, and primer designing for PCR, right? So why primer designing is important for PCR? Because PCR, let me talk about the basics of PCR first. So if this is our strand, so in PCR what we are having a, so let me take a color here. So let's take this one. Okay, we are having two strands. So let's say this is our DNA of our interest, and this DNA, first of all, it is first denatured. So now the DNA is denatured, so we are having two different strands separated now. After separation of two different strands, what we'll be doing, we'll be adding some primer. Here comes the application of primer here. So we'll be adding our primer. So here the primer is depicted as this pink color short nucleotide sequences. It's a primer. It's usually 18 to 30 nucleotide sequence long. It is usually 18 to 30 nucleotide sequence. Okay, so it varies from time to time from type of PCR we are doing for the amplification of a gene. And why we are applying PCR? Because we are amplifying the segment of a DNA. So suppose in this whole so there is a lot more section are also there in this DNA. So here is a section throughout this place, but we only want this light color section to be amplified because this is our desired gene, right? So for that reason we'll be adding the primer, which will be added at the terminal region. So here one primer adds here, another primer is added here. So this is two different primers we'll be adding. Now one primer is called a forward primer or FP, another primer is called the reverse primer or RP in this case. Okay, so we'll be adding two different primers, then they will elongate the gene of our interest, and finally what we'll get, we get our desired product. So that means one product will be this. So let me delete it. So here it is, two different products. So here we have our old strands, but now these are the new synthesized strands. So these are the two new synthesized strands, and we will be having our product like that. Okay, so this is our actual PCR process, right? Now in this process we need to add this primer. Why we will be adding this primer? Because we know that for any kind of polymerization of nucleotide sequences, we need polymerase enzyme, right? Polymerase enzyme or DNA polymerase, like that. Okay, so this polymerase that we are using, they cannot function until and unless they get a free 3' hydroxyl end. 3' hydroxyl is required, and this 3' hydroxyl must be free, otherwise they cannot work. So for providing them the 3' hydroxyl for free, we need to use primer. Because what primer is simple sequence. Let's say this is a primer, so it's a simple sequence. It is having a 5' end, and also it is having a 3' end. Now the 3' end also is having hydroxyl. So now when the primer is attached to a DNA template, which is here in this case this light blue template, now when it is attached, now you can see here this primer is providing the 3' hydroxyl, which is free to react. Now new nucleotide sequences will be added, and this strand will be extended in this direction, right? So that's the basic need for primer in PCR. Now for the designing of the primer is very important, because now there is a question. There is a primer length, we all know that it must be from 18 to 30 nucleotide sequences, but the question still remains, what kind of nucleotides we'll be using in the primers, right? And what will be the arrangement of the nucleotides? Let's say they are telling us only 18 to 30, so let's say we take only 20 to 20 or 21 nucleotide sequences. That's for fair, so we are taking only 20 nucleotide sequences for example. But we are taking, if we take only all the sequences like this A, so 20 A sequences like that, 20 sequences, whether it is going to solve our problem? No, because it will end up with a very bad primer. Why is that, right? So that means there must be some important criterias for the selection of primers. And what are those criterias? Those criterias are written over here. There are 7 different criterias that I am going to talk about in this video, and here is 3 different primer. Now I will be taking example of these 3 different primers and telling you using these different parameters, that among these 3 primers which one is going to be the better primer. So for a primer designer, obviously, who is setting the experiment, even you, me, and whoever is doing the experiment, they need to set the different primers using softwares. So we need to keep this 5, 6, or 7 different things in our mind before selection of the primers. But nowadays we are having softwares, automated softwares. They will do most of the job for us, but we need to think about that, right? Now let's begin with that. So first of all, important part is the primer length, because you can't take a primer of any length. So they provide us a range of, remember, they provided us a range of 18 to 30 nucleotide sequence long, but that doesn't mean you take just any kind of length. It will depend upon the sequence you're amplifying, right? Now usually it provides that usually the primers of very short length we will not end up with a good result. So we must have at least greater than 18 to 20 primer, 20 nucleotide sequence in this case. That's a take home message in this case. For example, say if these are the three primers. So this is say primer number 1, this is a primer number 2, this is primer number 3. And in these three primers, if we look at the first one, it is 21 nucleotide sequence long. This is only 10 or 11 sequence long. This is 22 sequence long. Now among them, we can see the second primer is very short. It's only 11 nucleotide sequence long. Now why we are telling that short nucleotide sequence primers are not good? Because let's say this is our desired gene, desired DNA for to be amplified. Now if we put our primer, so let's say it's a long DNA, right? Our desired DNA is long. If we put a short primer, let's say this primer is, which is a short primer, so let me take this primer as this color. So if you put this short primer, this can attach to any place. It can attach to this place or this place, because as it is a nature of short nucleotide sequence, it can find a complementary nature in this target DNA. So it can bind with many different unwanted regions. So when you choose a short primer, so short primer is giving you unwanted binding. So you need to be take care of this thing, because it ensures the unwanted binding. So you shouldn't take short primers, right? So this is the first criteria. So using this criteria, we can tell the second primer is not good. So it's one cross there, and this one is good, this one is also good, using this particular criteria. Now let's move on to the second criteria, which is the melting temperature. Now melting temperature is important. And what do you mean by melting temperature? Remember, in this process of PCR, which we have just discussed here, that first step is to denature this target strand. Then, so for this denature, we need to provide a huge temperature. The temperature is almost 90 degrees Celsius and above 90 degrees Celsius temperature. And after the denaturation, we should lower down the temperature so that primer can anneal at this place. This is called the primer annealing or primer hybridization. Now for that part, actually most of the cases what we use approximately 54-55 degrees Celsius temperature for the annealing of the primers. After that, we need to synthesize the polymerization action, and for the polymerization action we must increase the temperature a little bit, and we increase the temperature almost to 72 degrees Celsius temperature after that. And then this extension process usually occurs. Okay, so these are the three different temperature variation that are going on in PCR reaction. PCR is nothing but the change in temperature in the environment. It will do the job for you, right? So melting temperature is important. Say here, if in this case, another important concept is that we are choosing two primers, right? One forward primer, one reverse primer, right? So we need to think that what is going to be the melting temperature or hybridization temperature for forward as well as reverse primer. Suppose you design a forward primer which is having a melting temperature of say 55 degrees Celsius temperature. So or let's say it is having a hybridization temperature of 55 degrees Celsius temperature. That means when you put 55 degrees Celsius temperature, this forward primer is going to attach with your target DNA, right? Now let's say we are having another primer which is reverse primer, and the melting temperature of reverse primer is say 60 degrees Celsius temperature or 65 degrees Celsius temperature, right? So now this one is having 55, this one is having 65. So but what we know that for the carry of this PCR reaction, we must attach both the primers, forward as well as reverse, simultaneously. But one primer required 55, one primer requires 65. So if you provide only 55 degrees Celsius temperature, forward primers will only go and bind, but reverse primer won't be able to bind, because the optimum temperature for its binding is 65 but not 55, right? So as a result of that, what it will do, that only forward primer will bind, no reverse primer binding. So it will lead to very less amplification. So it will amplify only this strand but not this strand. So in each round of PCR cycle, you will end up with only one type of strand amplification, and as a result you will be having a very less concentration of your desired product. So that is giving us a very important insight of the melting temperature. So we need to take care of the melting temperature for two primers. So if we choose forward primer and reverse primer, so this is the take home message, that if you choose these two primers, you must take care of the important thing, that the melting temperature of them should not exceed between 5 degrees Celsius. So let's say the temperature for aniline for forward, if it is 55, then the resting primer cannot exceed 60 degrees Celsius. So you need to keep these things between the 5 degrees Celsius temperature range, right? So that's a very important concept. Okay, and now let's talk about the third parameter, which is the primer dimer. Now what do you mean by primer dimer? Now we are taking two primers. We are taking a forward primer. So let's draw this. We're taking a forward primer. Let's say this is our forward primer, or say FP of our interest. And let's draw a reverse primer. So let's draw a reverse primer. So this is, say, the reverse primer, right? So the reverse primer. This is a reverse primer, RP. Okay, so we take two different primers. So what do you mean by primer dimer? Primer dimer means definitely, is there any complementarity nature that are present in two forward and reverse primers? So there is a question, is there any complementarity? Now if your both the primers, forward as well as reverse, if they are having a complementarity sequence, for example, let's say here you are having a then G, C and T, and in this case you are having, say, A, C, G, A, C, something like that. Now if you are having these two sequences in forward primer A, G, C, T and reverse primer A, C, G, A, C, you can imagine here one important concept is that in all this case, here it is G complementary C, here it is having a C complementary G, and this is a T complementary is A. So you can find three consecutive nucleotide complementarity in the forward primer as well as in the reverse primer. So what it is going to do, it will bring this forward and reverse primer close to each other. So let me draw it right way. So it will bring them close to each other, and as a result it will start forming bonds. So they will be self dimerize instead of dimerizing with the target DNA. That is completely unwanted, right? So for preventing this, you must not take any complementary sequence between the forward as well as in the reverse primer, right? So that's another parameter. Now let's look for the fourth parameter, which is the GC content. Now this is very important. Now why GC content is important? Because what GC content is telling us, GC content is telling us something about the temperature for melting, or melting temperature, right? So if you're having high GC content, that signifies the high melting temperature. Why? Because guanosine usually pairs with cytosine via 3 hydrogen bonds, which is much more stronger than the bond that we usually see between the adenine and thymine, because they are having only 2 hydrogen bonds, right? So in a primer or in a DNA sequence, if there is a higher GC content, that means there are higher bonding or strong bonding, so we must provide higher temperature to melt them. If you are having more AT, we require less temperature to melt them, right? So what we need in this cases, all these cases, what we can see, that we are denaturing the DNA, we are melting, we're attaching the primer or hybridizing the primers, then we are again denaturing them and again hybridization. So we are actually playing with the hybridization and also annealing and reannealing and all these things in each cycle. So if we choose something which is having much more GC content, let's say all this primer is made up with only G and C, so it will be extremely difficult for us to usually kick that primer away, or it will be hard for us to what we can say, uh, denature those DNA sequences, right? And obviously if we choose more AT rich sequences, then it will be difficult for us to keep that primer attached. So in both the cases it is troubling, right? So it should be a very optimum GC content should be there, and the optimum content here, it is 60% of GC content, or over 60% of GC content is fine. 60% GC content is required. And if we put 80 or 90% of the GC content, it will lead to a devastating effect, because primer will sit onto the target. Even if you provide energy, even if you provide temperature, it won't be able to kick this primer out, right? And if you take more AT content, as a result of that, primer will be so unstable to bind with the target DNA, right? So we need to think about that. And if we look at this particular three different primers, we can find this first one is made up with mostly A and T sequences. It's almost 60 or 70% of AT sequence, right? And 30% of GC. only 30% GC sequence like that. So it is not going to provide us a good primer because it won't be able to bind with the sequence pretty well. Right, and this one is very short and it is also having distributed G and T, so it's not a problem, so it's good, but it's a short one. So uh, we eliminate this one at the very first place because it is short, but still using this parameter of GC content it is good, so we put it right here. But this is GC content, this one is not good, so we put across. But this last one we can see this is a lot more high GC content. As you can see, it's almost GC contents, it's almost 65 to 70% of GC content, so it provides good a good result. So we'll put a tick here or right here. So this will be the assessment based on the GC content. Now let's look at the fifth parameter, that is the runs and repeats. Now what do we mean by runs and repeats? That means the repeats that are present in the primer. Now as the primer is a nucleotide sequence, there can be repeats. So repeats, as we can see here in this first one, we can see repeats like AAA, TTT, and you can say CA, CA, CS. It's almost repetitive sequences, all these cases, all repeat sequences we can find. Now these repeat sequence are not good. Why they are not good? Because if you are finding repeat sequences, there is a tendency for the primer to self dimerize with each other. So simply, if these primers are having a lot of repeat sequences, it will self dimerize it with itself like that and form a secondary structure, and we don't want this, right? And obviously if there are a lot of repeat sequence, there is a tendency that in this target DNA somewhere, so let me take here, if this is a target DNA, somewhere in this target DNA, somewhere here must be there is a AAA sequence or CACA repeats. So it's a tendency for all these primers to come and bind with this sequence, right? So it's almost tendency they will come and attach to this unwanted region. So lot of repeats and runs will provide us unwanted, again unwanted or undesired product. So we don't want this, right? So that's another important assumption. So based on these runs and repeats, what we can see that this first one is having a lot of repeats, so this is not good. The second one is not having that this is not having that much of, it's also having say CC, GG, and TT and all this repeats, so it's also not that good. But this third one we can see, though it's having a repeat like GG, one small single repeats, but it is uh, almost good. So this one is really good based on the parameter of runs and repeats, right? So now let's talk about another two. So here we go, the last two different parameters, and here it is, the distance between the primers. This is also a very important and key point. Now what do we mean by the distance between the primers? That means obviously the length of the DNA sequence we want to amplify. Let's say this is the DNA sequence, so let me take a color here, let's say yes, this one. So let's say this is a segment of the DNA and this is a stretch we want to amplify. And if we put the so let's say this is one strand, this is another strand, and if we want to amplify all of them, so let's say this is one primer, binds here, and this is another primer, it binds here. In both these cases there should be a difference between these two primers. And what do you mean by the difference? Here it is, the difference is from this to this direction, so this part is the difference between these two primers, and this should be uh, at least 150 base pair, or at most, so it is at least 150 base pair, and at most 110 kb or 10 kilo base pair, it is at most, right? So this is another very important assumption. So the desired gene of our interest or the target DNA, it must be 150 base pair long at least, and obviously it shouldn't be more than 10 kb long. Then we can't use probably the primers, because in those cases, if this is a large distance between these two primers, there should be the chances of misbinding of the primers in somewhere in the middle of our target or amplifiable DNA, right? So as a result it will lead to the inefficiency of our product, right? And obviously if this is a very small distance, then the primer will self dimerize and it will lead to bad result. Okay, so that's the important point. And the last point is the secondary structure. Now secondary structure is also important, because if we're dealing with all these important stuffs, the secondary structure means RNAs and DNAs, or nucleotide sequences, whatever they are, they can form structures. Because like this one, this is a kind of secondary structure what I have drawn. Let's say there is a similarity, there is a complementarity, a self complementarity that are present. Let's say in the same primer we are having C, G, A, T, C, and obviously let's say T, A, G. So if this kind of sequence present there, so what will happen there, you can find that there is a complementarity nature, that this G can bind with this part. Let's say uh, say G, and say, so let's say here it is A, here it is T, like that, and C. So here let's say uh, there's a T, A, and G, so like that. So if let's say this is, if this is a primer, then the probable chance of folding this primer in such a way so that it can find the complementarity. Where is the complementary? I'm telling you. Let's say here this is a G, so here it is a G, here it's a C, complementary of each other. T with A with this T, and G with this C. So these are the complementary. So we can find if it folds like that, if it folds like that, it can form what we call a secondary structure, and this will often lead to the formation of this kind of structures. This is called hairpin loop, or sometimes it will lead to the formation of this kind of structures, like this kind of structure, which is called a stem loop structure, and all these structures that are found in RNA sequences and also in DNA sequences. So this kind of secondary structure must be prevented. So we choose such nucleotide sequences to build our primer, so that they shouldn't have any kind of self complementarity nature in them. So if we find here in this case, this one is having repeats, and it lead to the self complementarity. This is a very short to have any self complementarity, so we'll better write here in this case. But this first one can have self complementarity, so I will be putting a cross here. And the last one will, it cannot have any complementarity, because it is well distributed, so it's a tick for that. So using all these parameters, if we provide the marking to these three different primers, what we get, who is the best result? The first one is getting one right and four wrong, so it's a 1 out of 4. The second one is a 2 out of 4. The third one is a 4 out of 4. So it is telling us that this third one, the third primer here, it is the best primer for our PCR among these three primers, right? So these are the different techniques and tendencies that you should take care of when you design the primers. And I hope that's helpful. Thank you.